Okay, excellent. Okay, let's get started. So today we will finish the last, but another least part of the building blocks of the life, that is the plasma membrane. And then we will talk about some very important, very fundamental phenomena in the cell, and also the associated mathematics derivations. Okay, that is we will discuss about the probability, statistics, and the random walk, and those will give rise to the very important phenomena such as diffusion, but there are many more. Okay, so in terms of announcement, we have the homework number 1 due today, so you can email me the homework in the PDF. I received some of you, many of you actually, the homework number 1 in the email. That's great. Yeah, just send it in an email and then I will get back to you. Okay, so I briefly looked into some of the students' homework number 1. It seems you guys are doing well. So now, after homework number 1, we will get more homeworks that will be more quantitative, and then we involve MATLAB. So I have the MATLAB code, MATLAB tutorials uploaded on the Canvas. You can learn from there if you are not familiar with it. Okay, and I do encourage you to form groups to study, and then to learn from each other. So from homework number 2 on, we will have more quantitative practice, and then you can use MATLAB to solve many questions. Okay, so that's a very powerful tools that you can use. This Friday, we will have a midterm exam number 1. It will be in the class, and as we explained, it will be containing 10 questions, and I have put some example questions online already. So it will be open book, open notes, and all the questions will cover the material we will finish before Friday. Okay, so today and Wednesday's class, the materials are also important. Okay, good. That's all for the announcement. So I hope you all enjoy the class and focus on the science in our materials. Okay. Okay. So if there are no other questions, let's start our today's material. Can you guys hear me?
Yes.
Yeah. Yeah. Excellent. Very good. Very good. Good. So last but not least, the building blocks we will cover. That is the number 4 is the lipid. So on the right hand side, that's a picture showing the lipid. Okay, so here actually is the biolipid layer. You will know why it's called biolipid very soon. So this lipid, you can see it. This actually is the cell. This forms. So the lipid forms the membrane. Okay, so this is actually a membrane that separates the cell inside from outside. Okay, so there is inside the cell part and outside the cell part. Okay, and as we discussed before, this lipid is not just an inert material. It also carries electrical charge, and this charge forms an electrical field on the across the membrane, right? And also this lipid also carries the mechanical force. So there is a mechanical field and electrical field across the membrane. Those act together to influence the energy landscape of the membrane, and then those energy change can alter the conformation of the transmembrane protein, and then that trigger many interesting chemical reactions and biochemical processes, right? So yes, lipid is very important. All the 3 previously discussed macromolecules, they are actually defined by the chemical structures, The lipid actually is not a polymer. It's more defined by the physical properties. They are made of hydrophobic and hydrophilic components together, and overall they are actually, because they have the hydrophobic components, so overall they are insoluble in the water. So this lipid actually is widely used to form the structure, not for energy use, for structure use in the cell. And then because they are insoluble in the water, so they provide a really good barrier to prevent certain ions and cations and the different ions that are harmful for the cell. So the lipid can form those barriers to protect the cells. Okay. There are three major types of lipid in the cell. Major types. There are other smaller types as well, minor types as well. Okay. So this is the most commonly found lipid in the cell. Those three are triglyceride, phospholipid, and steroid. Okay, and then though they individually they actually can store some energy, but that's not their major function. They mainly form the membrane, and then on the membrane they can transduce signals, and also because, as we said, cells can form the membraneless organelle in the cell, right? Called to form those membraneless organelle of the protein that perform certain functions. Cells in the same time, cells can also use membrane to quickly assemble different face to have the organelle with membrane to be formed in the cell and then perform function. So cell actually can have many different phase separation occur in parallel in the cell. Okay, so those are actually very useful functions. Here is the cartoon shows the monomer of the lipids. So this is a fatty acids. We have three types of. So this for the lipid they have the several different monomers. So this is the tail part. They have here shows three different tails. Okay, these tails can either saturate. Those tails are made of carbon atoms, and then they form the polymer chains. When they are saturated, means they do not have a double bond. The tail only have a single bond. Then they are strict. But if they happen to have a double bond, it means they actually contain energy, right? Extra energy. So if they have a double bond among these 19 carbons, they have one double bond there. Then this chain will get bended. They will bend. Okay, so they are not strict. And for the unsaturated bond, in fact, they can release the energy by breaking this bond, and then they will straight up again. In the cell, those they are not static. All those monomer actually can freely jump up and down due to the diffusion. So if we have a unsaturated bond inside the membrane, they actually can move around. They can move around. If you label it with certain fluorescent dyes, and then you can see how that move within the membrane. Okay, so that's actually very very useful way to transduce the signals. And those fatty acids can combine together, can align together. All of them form the covalent bond with another monomer, with another component called glycol. So glycol, we all learned this from the basic chemistry. Right? Glycol has 3 OH components, and then they can actually respectively carry out condensation reaction with the fatty acid. Can be different types of fatty acid. Okay, and then they can form one bigger monomer, and this covalent bond is called ester linkage. Remember, in the protein we also have different name. Although they are all covalent bond, and they are all through the condensation reaction, but they are due to the difference of macro molecule. They have different name. Peptide linkage and ester linkage. Those are for the protein amino acid chain, and the lipid chain, right? So this is ester linkage. They are different names. So here, when they form, when the glycol and the 3 fatty acids combine together, through the condensation reaction, they will form a triglyceride molecule. Okay, so that's a fat. So this is the one type of lipid, and then there are other kind of reaction as well. So glycol also can connect with two fatty acids, and then at this time they can add another group. That's the phosphate group, and then these three together form another types of lipid. It's called a phospholipid. So here actually is 4 different representation of a phospholipid. So phospholipid, you see they have a polar head that carries the phosphate group and glycol. And then it also has a non-polar tail. So here polar means they have electrical inhomogeneous or heterogeneous distribution of the electric charge within the molecule. Okay, for the tail, it has a non-polar tail that has a uniformly distributed electric charge, and in this case they are all saturated. So they are very straight, 2 tails, very straight. Okay, two chains. This kind of structure actually they are hydrophobic, and for the head is a polar, and this actually is hydrophilic. So this single molecule actually has 2 different properties on 2 sides. So they can either contact with water or contact with oil. So they can communicate in both ways. So this is the chemical formula of phospholipid, and it can also be drawn in terms of the schematic way, and also using the spacing filler. So here actually every ball is 1 atom. So this is a very common way to represent macromolecule in the molecular dynamics simulation. So this actually is the representation of phospholipid. Okay? And to be simple, the number 4 representation is using the icon. So here, once we see this, you will know this is the hydrophilic end, and the 2 tail actually is the hydrophobic end. Okay? And then here, phospholipid actually has many other different examples, different variants. So here shows 6 different types. For these 6 different types, they have different hydrophilic head, and have the same hydrophobic tails. This happens very common in the nature. Okay, so remember we talk about the difference between human cells and the bacteria cells, right? So bacteria is a single cell entity. Human cell is a multi-cell. In fact, even for the membrane of the bacteria cell and human cell, membrane composition also very different. So they have different phospholipid in the bacteria. So because membrane composition is different, that cause membrane properties are different. So the human cell, any membrane protein, any ion channel on human cell, it cannot be easily injected, embedded into bacteria cell, and vice versa. So the membrane protein, when people study their properties, if it's a human cells protein, you cannot put it into the bacteria cell body to study. And bacteria cell usually is sometimes they cannot produce these types of protein for when you do the protein expansions. Okay, so we have to choose when we study any transmembrane proteins, we have to choose their nature, membrane environment. Otherwise, the property we studied could be very, very different. And as we just said, the lipid has 2 different properties. Actually, it's 2 contradictory properties. One is hydrophilic, another is hydrophobic, and it has both together in one molecule. So this kind of molecule is actually called amphilipathic. So it has two properties together. So here shows this one molecule. If we have many molecules together, then because they have two properties together in one molecule, so in any solvent, they will only one end will like that solvent, not both. So then, if we have many of them together, they actually the phospholipid can aggregate together. That's the professional name is called self-assembly. So they can self-assemble together to let the final macromolecule behave to appear to be like that solvent, and to make the non-like component to be shielded inside the macromolecule. So that's what's shown on the right-hand side. So if we have many of those molecules put in the water, then their tails will be shielded inside the assembly. Okay, so those are long tail, they are hydrophobic, so they will get together to be in the center and in the core. And then outside, they will expose the hydrophilic part towards the water. So those are round the head, polar head, actually hydrophilic, right? So they will tend to contact with water. In this way, this kind of structure actually minimize its free energy with the environment together. So they are this is the most stable conformation for this structure. Okay. And if in the oil, then they will go in opposite way. So the hydrophilic part, that is the polar, right? Hydrophilic, they will not like oil, because oil is non-polar. So they will get inside and then let the polar, non-polar part to be outside. So the tail will be outside. So you can see the top one and the bottom one, they are very different. And then also in the water and oil interface, the polar part will go contact with the water, okay? And then the non-polar part will go towards the oil. So then they will form a monolayer here, okay? So in the many chemistry industry, they actually use this property to produce those macromolecules in a batch, okay? And then they also use this alignment, this well-aligned shape at the water-oil interface to make many of these monolayer. They can make a multi monolayer by repeatedly produce water-oil interface, and then many of these monolayer can be aligned together. Okay, just using this unique property. Okay. So yeah, that's the self-assembling I mentioned. Here is a diagram shows how the self-assembling starts in the cell. Okay, so here we have the purified phospholipid single molecule, and then they have the red hydrophilic part and the blue hydrophobic part. And then because in the solution in the body, in the solution, everywhere is water, everywhere is a solvent. Okay, no, not oil usually. And then they actually will form double layered. It's called a bilayer. It's called planary phospholipid bilayer. They form a bilayer. This bilayer is having the exposing the hydrophobic part inside. It expose the hydrophilic part outside, and then hide hydrophobic part inside, and then they form this bilayer. If it's a cell, then they will self-assemble, enclose themselves. Okay, enclose themselves, and then form a ball. This is called a lipsome. Okay, and inside is water, outside is water, and then in between those are the phospholipid bilayer. And for this process is actually an energy minimization process. If they are showing this planary bilayer, then there's still some hydrophobic part exposed to the water, right? So that actually is not favorable. So they want to minimize their contact with water. So that's why they curved together. They can reduce they reduce the chemical energy by connect the hydrophobic part together to avoid their contact with oil and sacrifice the bending energy. So they actually generate a ball that actually increase the bending energy, the mechanical energy, but that energy is smaller compared to the chemical energy here. Okay, so the overall is still an energy minimization process. And if you take a look at the cross section of this liposome, it looks like this. It is a very well aligned bilayer structure. So if the cell, so this is called a liposome. If the cell is behaving like a liposome, then as we know, the bilayer inside is hydrophobic. It will not like water to pass through, right? So then this membrane actually will not be very friendly for the exchange of the molecule or the ions to between the inside of the cell and the extracellular region. So as you see here, if the membrane is this pure, if they are so pure, all are made of phospholipid bilayer, then the large uncharged molecule will get blocked to penetrate through the membrane. They are rejected, like glucose or sucrose, they will get rejected. And also the ion, many ions, they have a charge there. Okay, even some of us small, but they have a charge. And then because they have charge, then they will have a hard time to penetrate through this hydrophobic inner core of the membrane. So this actually will disable the cell to have more nutrients and have more signaling molecules. Only small uncharged polar molecule or some small hydrophobic molecules can barely go through. Okay, so then we lose a lot. So that's actually is not true in the real nature. So in the real nature, the cell actually generates many transmembrane proteins, transmembrane ion channels that can selectively let all those ions to go through, and can also exchange glucose to let those larger polar molecules can go into the cell, not through the membrane, but through the transmembrane proteins. Okay, so this actually is what I'm just saying. Actually, is the inspiration that triggered people to study transmembrane proteins, and then that eventually, several decades ago, that eventually leads people to discover a water permeable channel in the membrane. So that's actually the Nobel Prize several decades ago. So it's based on the membrane's biophysical property, and then people find in the cell there must be some functional ion channels that can help the cell to exchange water and exchange other proteins. Then people studied the membrane more carefully, and then developed the many physical tools to study membrane, and then they find those ion channels. That is the Nobel Prize. And those ion channel actually play important role in the drug discovery, because many drugs actually use ion channel as a target to treat the diseases. Okay, so that opens the field. Now those membrane not only have the transmembrane proteins that enable the ion exchange and also enable cell-cell contact. So here is one type of protein that is called cadherin that enable the cell to contact with neighbor cell. But also those membrane actually contact with the cell cytoskeleton through the transmembrane proteins. Do you guys hear me?
Yeah. Yes.
Yeah. Okay, very good. So those membrane also contact with the cytoskeletons because the membrane itself, as we said, is only 5 nanometer to 10 nanometer thick, right? And it is very flexible, like the gel, like the hydrogel. Okay, if you look at it, it's very like hydrogel. So by itself, it cannot sustain the cell structure. So the nature developed multiple transmembrane proteins that those proteins can talk to the membrane as well as to the cytoskeleton. So they connect the cytoskeleton with the membrane. That enable cells to generate tension, membrane tension. Okay, generate tension on the membrane. That is make the membrane like a tent. Okay, then cell actually use that membrane tension force to sustain the structure of the cell, and also because the membrane carry out that tension, it functions similar to the cytoskeleton. Remember, we have derived how to measure the tension inside a single cytoskeleton filament. We derived that previously, right? We apply the force perpendicular to the cytoskeleton and then measure the tension force, right? So as we said, cytoskeleton use that pretension to transmit mechanical signal and then to maintain the cell structure. Membrane actually works in a similar way, but it's a 2D surface. Membrane is a 2D surface. It also maintain a membrane tension and then use that tension to transmit mechanical signal and then use that tension to maintain cell structure and also use that tension to regulate the conformation of the transmembrane protein within it. Okay, so measuring membrane tension is also a very important topic in the biology. Okay, so how can we measure tension in the membrane? How can we measure the tension in the membrane? We measure the tension in the cytoskeleton, but how can we measure the 2D plane? What's the tension in there? How can we measure? This can be a good question for all of us. So you can think, is there any way you can develop to measure the tension in the membrane? Is there any way? Okay, so that's a good question for everybody. Here, because due to the time limitation, I will introduce you one way that people measure the tension within the membrane. In the next lecture, after class, you can think about other ways, and then we can discuss about this in the next lecture. If you have a new idea, you can also measure the membrane tension new way, different from what I will discuss. Then that's great. Okay, that will be even better. So one way to measure membrane tension is use a tool called fret. This did you guys hear about the fret before? Do you know the fret before? Anyone of you know this mechanism? Can you explain to me what is the fret?
Yeah, basically it's kind of similar process as fluorescence.
Okay.
When the excitation or the emission and excitation of two fluorophores overlap, there can be some non-fluorescent resonance energy transfer, which excites the second fluorophore, and which then releases photons as fluorescence.
And then what is your readout?
The readout?
Yeah. What did you read?
Like what signal you get at the end?
Yeah. What kind of signal is useful for you?
I guess I see what you're saying. I see what you're getting at. I think it's often used as like a I think they call it like molecular ruler because it's inversely proportional to the distance between the particles to raise to like the fourth power or something. So you can get pretty accurate measurements in like nanometer scale based on the distance between the particles.
Good, good. Jackson, you give a very overarching description of the FRET. I think that's very good. Let me re-explain it in a more detailed way. Can everybody understand what Jackson was talking about? Do you guys understand what Jackson was talking about?
I'm still a little bit confused.
Okay, good. Let me explain it to you and to all of you. And then if you have a question, ask me immediately. So here, FRET actually contains, is called a fluorescent energy transfer. So regardless of this name, it contains three things. FRET contains three things. It contains two of them are fluorescent protein. Two of them are fluorescent protein. Third one is a molecular linker. Molecular linker is a peptide linker that links these two fluorescent protein in between. So let's look at it one by one. For the fluorescent protein, we will talk about more detail about the fluorescent mechanism, physics, real physics mechanism in the next section when we talk about the microscope. Okay, here I just briefly mention the mechanism of a fluorescent protein. So a fluorescent protein is a protein that can emit out fluorescence, and that fluorescence is has a certain wavelengths. Okay, certain wavelengths. But for the fluorescent protein to emit out that fluorescent light, it has to be excited by another light first. So usually fluorescent protein actually need to receive a light has a short wavelength. Short wavelength means high energy, right? So the fluorescent protein need to receive a short wavelength light and then emit out a fluorescence light that has a longer wavelength. That means it actually conducts energy transfer. It takes some energy from the light, higher energy, and then emit out another energy that is less energy. In between, there are some energy loss. That energy loss is actually generate heat in that fluorescent protein. Okay, this part is understandable or not? Is that a part understandable?
Yeah, that makes sense.
That makes sense. Okay, and then for the fluorescent protein, different fluorescent protein, they actually accept specific low wavelength light to be excited. They are not excited by all kinds of light. They actually accept a specific wavelength light to be excited, such as the green fluorescent protein. They actually accept the wavelength in the blue light region, 400 nanometer, and then they emit out green light. That's why it's called a green fluorescent protein. If it's a yellow fluorescent protein, then it actually accepts the light in the 500 nanometer region. It accepts green light, and then it emit out yellow light. Okay, so that's why it's called yellow fluorescent protein. Okay, so it's not everywhere, all kind of protein. Now, the FRET, first two components, the FRET actually contains two carefully selected fluorescent protein. These two protein, this is, as I show here, the blue one and the green one, these two, they are carefully selected in a way that this blue one, the fluorescent, this guy, when they emit out, when they send out fluorescent, it send out the fluorescent light sent out by this guy can be exactly the one that can excite this green one, and then this green one can emit out the light. That light could be yellow. Right? In the spectrum, in the light spectrum, high energy to low energy is purple, blue, green, yellow, orange, and red. Right? So far, is understandable or not?
Yeah, that makes sense.
Makes sense. How about other students? Don't be
Yeah.
silent.
I think it's fine.
So far is fine. So far is fine.
Yeah.
Anybody has a question? Because I'm going to one step deeper now. Everybody understand me? Okay, good. Nobody, everybody understand. Okay. Now, when these two fluorescent protein are put together, they if they are far away, spatial distance, if they are far away, when this blue one emit out the light that can be used to excite this green one, if these two proteins are far away, then that light emit out is light is going in the volume way, right? Radially 4 pi, right? So they can quickly lose their concentration, right? Lose their dosage. If these two are too far away, then the light emitted out by this guy, by this blue one, cannot excite this one, cannot excite the green one. Then what do we see in the microscope? What do we see actually is the green light emitted out from this blue one. We see the green color, okay? But if this green fluorescent protein is close, very close to the blue one, then the light emitted out from this blue one actually can excite it. Then all that green energy, green light actually is absorbed by this green fluorescent protein and then converted by it into the yellow light and then emit out. Then what we see in the microscope is yellow light. Okay, so this is a very unique distance dependent phenomenon between two fluorescent proteins. This part so far is understandable. So far, is no confusion, right? Good. Now, based on this property, then people actually put the third components in now. So they put a peptide in. This peptide is from the spider web. Okay, so spider web has a very elastic. Spider web is very elastic, not plastic, not viscoelastic. It's very elastic. So very elastic peptide protein. Then they put a peptide in between, connect these two. Then that peptide will work like a spring. If you apply the force to if you apply tension to these three components, you stretch the blue one from the green one apart, and then you add the excitation light, maybe purple light, to excite this blue one. Then this one will emit out green light, and then that light will not be absorbed by the green fluorescent. Then we will see the green light. Then that means this components is under the tension, and that tension is large enough to let these two proteins to be far away from each other, and then there's no light energy transfer between them. Then we know there's a tension. If there's no tension or there's a compression between them, then these two proteins will get very close, and then this spring get stressed, right? Spring get compressed. At that time, if you excite the blue fluorescent protein, the emission green light will be absorbed by the green fluorescent protein. Then this one will get excited and then emit out the yellow light. We will see the yellow light. Okay. And then by knowing the relationship between this green, yes, this here is green. because there are no transfer. This is green because they're too far apart. Only green light from this guy, right? Green light. The green to the yellow, they have a range change. The color has a range change. We can measure corresponding distance between the fluorescent protein, and then that corresponding distance is also related to the force between them, because this peptide from the spider web, we can measure their spring constant, and F equal to Kx. Once you know the spring constant, then between the distance, you can measure the force, okay? And measure force. Then this three component device, this is molecular device, can actually be used as a readable spring, can be injected, can be inserted into different part of the cell, and then measure the force accordingly, right? So here is the most successful application of this Fred device is actually on the membrane, on the cell membrane, and on the protein. If there are big protein that can change their conformation, this molecular tool can be used, can be bonded to the bigger protein, and then find out the color change, the red to the green shift or green to the red shift during the big protein conformation change. Okay, so in the protein and in the membrane, they have been successfully used, but they are not very successfully used in the cytoskeleton. So that's why if you measure cytoskeleton tension, you cannot use this one, mainly because the cytoskeleton conformation change is not that big. So they cannot easily excite the green to blue, green to the yellow color change. It's very, very subtle. So it's not that efficient. Okay. So here is one paper, very recent. This is actually the first success to use this device to measure the membrane tension. So people used this. You see this pair. They added the membrane bonding domain to this pair, and then let those membrane bonding domain bound to the membrane, and then so this is the membrane, and then they have this device inserted into the membrane. This is like in engineering, it's a bit like the strain gauge, right? Strain gauge. So then when the membrane get deformed, they will pull those membrane bonding domain, and then pull these two fluorescent protein that will also in turn pull the spring in between. Okay, once the spring in between is pulled, then the distance between the fluorescent protein pair changes. Once that distance changed, then when you shine the purple light to excite this guy, the emission you collected will change. Okay, here is one control experiment. This actually is a control experiment. So in this way, in this one, the people only bound one fluorescent protein with the membrane bonding domain. Another one is not. So then whatever membrane get deformed, they will not influence the distance between the two fluorescent protein. So there will be no change, right? So they put both this control and the real protein inside, and then apply force to the cell, like here, shear force to the cell. See whether you can see the change or not. In theory, this one will show no change. Then that means it's correct. And then any change of the color here tell people what there is a force difference. And then based on the color, you will know bigger membrane tension or small membrane tension, right? So this actually the people have validated it works. So you can see here is in the cell, they find they actually inject many of these pair into the membrane, and then you can see the distribution of the color on the cell at the different regions. Okay, so here you can see membrane tension actually is not uniform. It's actually patched, right? So different region of the cell, they show different color. They have different membrane tension. Here is they apply the shear stress at 0 minute at the beginning. There are some rest tension, rest tension, because you know the cell generate the tension through their cytoskeleton, right? And then after 15 minutes, the shear stress apply, external shear stress apply, the tension change, they change color now, right? And then the control is this one, KMS. This is the control. This is that is this guy. Shows only one end is embedded in the membrane. Okay, so you see before and after there's no significant change. Okay, for the others at a different value of the shear stress, look at the raw, different raw. They are stand cell. Okay, they all change the membrane tension. So this is actually a very good way to measure the membrane tension. Okay, but this method also has their limitation because the spring here cannot be stretched infinitely. So there are only a small range of the force you can measure. So in the field, people still need to get a new method to measure the membrane tension. Okay, so it would be great that you guys, all of us, think about the other way, any other method that can be used to measure the membrane protein, membrane tension. Okay, good. Is that clear? Is that clear? Yeah. Okay. Good.
Dr. Tang,
Yeah. Go ahead.
I have a question. So for the fluorescent protein, the blue one is excited by the purple light and then emit the blue light, and the green one is excited by the blue light and emit yellow light. Is that?
It depends. Good question. It depends on the protein you choose. So when the fluorescent protein is excited and then emit out the corresponding fluorescence, there is a phenomenon called a Stokes shift. So that means the input wavelength is always smaller than the output light wavelength. Okay, so in this case, people choose that the emission of this protein. You see my mouse or not?
Yes.
The emission of this protein is sufficient to excite this protein. It's sufficient to excite this protein. Okay. And for any protein, when they get excited, they will emit out the fluorescence that has the wavelength smaller than the excitation light's wavelength. But depending on the protein, different protein, it's a exact wavelength number can change. Okay, so as I heard you said that the blue light, purple light in and blue light out, right? You said that. Sometimes depends on the protein types. Sometimes it's purple light in, but the other light is between purple and blue. It's not completely blue, depending on the protein's properties. Okay, but it's always the light emission wavelength is larger. Okay,
got it.
For one photon. By the way, there's one exception actually. What I'm talking about here, the Stokes shift is a one photon get in and then one photon get out. There are many photons get in, and then correspondingly, every time a photon means the single unit of the light energy. Well, every photon get into the fluorescent protein, that is converted into another photon coming out. That coming out photon has a longer wavelength. Okay, I'm saying that. You hear me?
Yeah, that makes sense.
But there are also exceptions. There are two photon case. If you have two photons that has a longer wavelength, if they hit the fluorescent protein at the same time, their energy actually can be added together. They added together and then used by this fluorescent protein and then converted into another photon to emit out. So two photons in, one photon out. At that time, the wavelength this guy send out is shorter than the wavelength coming in. So that's why in the two photon microscope, you use a red light to excite the protein. And then that protein can combine two photons together and then emit out the green light. Okay, so this is case by case. Okay,
Thank you.
Sure. Okay, very good. Right now it's 3:50. So we will discuss about the diffusion and mathematical derivations in the next lecture.